deeper understanding of cell function through the use of multiphoton intravital microscopy. We're really pleased to have as our guest presenter this month Dr. Torsten Mempel. Dr. Mempel is at the Massachusetts General Hospital. He's the principal investigator for the Center for Immunology and Inflammatory Diseases. He's also involved in medical education as part of the Harvard Medical School system. And I think most significantly, with respect to this webinar today, he has over 10 years of experience in the use of multiphoton intravital microscopy to study immune system function. The agenda is I'm going to give a brief description of the system that Torsten uses, and then turn the presentation over to Torsten. At the end, we'll have a Q&A session. So the system in Torsten's lab is a Bruker FM Ultima in vivo system. It's built around the Bruker FM in vivo microscope platform. Uses standard raster scanning XY galvanometers. It's a dual wavelength excitation system, so he has two IR lasers integrated with the system. One of them is a MiTi HP, and the other one's a MiTi Deep Sea. 4 channels of detection using multi-alkali detectors, motorized focus, and then the 2 objectives he works with, the 20x 0.95 ANA and a 60x 1.05 ANA. So without further ado, I'd like to turn the presentation over to Torsten.
Well, thank you very much, Jeff, and welcome everybody. So as Jeff mentioned, we use multiphoton intravital microscopy to study T lymphocytes functions primarily. And just to give you a little bit of background to our studies, so as the immunologists among you may know, T cells are very migratory cells in the adult mammalian organism. And what we study is how they migrate between different tissue environments, like the thymus, peripheral lymphoid tissues, or non-lymphoid tissues, and how they interact there with different types of antigen-presenting cells that provide them with instructive and selective signals that determines their differentiation into effector cells, and that ultimately also control the execution of these effector functions when, for instance, they infiltrate a tumor in a cancer patient, or when they engage with virally infected cells in peripheral tissue. So basically our main approach to study these events is direct in vivo visualization, and that's what we use our microscope for. And I will be speaking about 3 topics today. First is some general studies we do on the migration of cells in tumor tissue, focusing on effector and regulatory T cell populations and how these functionally interact with each other. The second one will be on a related subject, again, CTL, cytotoxic T lymphocytes infiltrating tumors and rejecting them, but in this context, our efforts to visualize signal transduction events in these cells at subcellular level. And in the third and final part of my talk, I will be talking about, so to speak, the flip side of cell migration in the immune system, which is that certain pathogens have probably learned to take advantage of the migratory behavior of lymphocytes and other hematopoietic cells in order to disseminate within hosts and create systemic infections. And the pathogen I will talk about is HIV. So we study the behavior of HIV-infected T cells in lymph nodes of humanized mice. And then hopefully, as Jeff pointed out, hopefully we'll have time for questions, but please feel free to raise your questions as we go along. So just very briefly, again, the background to our first study. So then a cancer growth, malignant growth arises in the body. This induces the effector cell populations, which through their various effector activities, cytotoxicity, cytokines, can actually control or even reject these malignant transformed growths in many cases. But both the tumors as well as the effector cells also induce regulatory immune response components, including the activation of regulatory T cells. And these regulatory T cells have multiple mechanisms by which they can antagonize effector cells, which are, however, not very clear. And what we are trying to understand is how is the information between the regulatory T cells and the effector cells, the information exchange facilitated. And the model system that we use for this here is that we take mice, regular biopsy or black 6 mice, inbred mice, and we implant those with tumor cell lines. In this case, this is an epithelial tumor, CT26, that is genetically engineered to express a surrogate antigen, hemagglutinin. This is actually influenza hemagglutinin, but in our case, this serves as a surrogate tumor antigen. And we also express fluorescent reporters in the tumor cells, in this case, a fusion protein between H2B and histone protein and the blue fluorescent protein cerulean. Then on the same day, we infuse these mice by intravenous injection with regulatory T cells that express the red fluorescent protein TD tomato or other fluorescent reporters, GFP for instance, and that are specific for hemagglutinin. So these express a transgenic T cell receptor that specifically recognizes the tumor-expressed hemagglutinin. And what happens is that these cells migrate to lymphoid tissues, spleen, lymph nodes, and in the tumor-draining lymph nodes, specifically these cells accumulate, they proliferate, and become activated, and eventually, like normal effector T cells, they leave the tumor-draining lymph node and start to infiltrate the tumor tissue. They further expand by proliferation, but also by enhanced survival, we think, in this tumor microenvironment, and now change the immunological conditions of the tumor microenvironment in ways that we want to study. And now, if we come into the third cell population, these are hemagglutinin-specific cytotoxic lymphocytes. So these are the cells that have the capacity to eliminate these tumors. These cells directly migrate because they're in vitro primed, directly migrate into the tumor tissue, and in the absence of this HA Treg, they actually reject the tumor, but in the presence of the HA Treg, the tumors continue to grow. So we have what is called tumor tolerance. So now this is what we are started with. And what we are using as a technical approach is that these tumors, we implant in the so-called dorsal skin fold chambers. That is basically the dorsal skin flap of a mouse, which is sandwiched between two titanium frames. The skin is excised on one side, so we can implant the tumor into subcutaneous tissue on the other side of the skin flap. We cover this with a glass cover slip, and this way we can longitudinally observe how the immune response unfolds in the skin fold chamber in the tumor over time. So you can image the same dorsal skin fold chamber over the course of several days and weeks and see how the tumors T cells infiltrate. And we then intermittently again anesthetize these mice, put them under our Two-Photon microscope, and record stacks of optical sections like you would do on a photo microscope, just that we do it iteratively, typically with time intervals of 15 to 13 seconds. And then we display these, after we render them offline into three-dimensional volumes, we render these stacks of optical sections and then display them at 50 frames per minute. So we accelerate everything typically at 225 or sometimes higher speeds. So this is now just an example of such a recording. So here you see in blue the nuclei of the tumor cells. White are all the blood vessels, which are the larger blood vessels are here in the so-called tumor stroma. Those are the tissues that surround the tumor. And here in green and red you see the specific CTLs and the regulatory T cells. And what we're interested in here are various aspects of their behavior when they see their antigen or they do not see their antigen, and ultimately we try to understand how they interact with each other. We know from this recording, for instance, that they don't directly physically interact with each other, but there must be other indirect means by which they communicate, because we do know that they do communicate. So one of the questions we've been trying to address, for instance, is what are the cells with which HA-specific regulatory T cells functionally interact in the tumor. So what kind of cells present cognate antigen HA to these Treg to make them do the things that they otherwise would not do? Are these dendritic cells or more macrophages or the tumor cells themselves? And to address this question, here we have a mouse where we infuse GFP expressing HA-specific regulatory T cells. We label the blood vasculature, in this case by tritsi-dextran infusion, so that's a large molecular weight plasma marker. And again, we have here our H3 cerulean expressing tumor cells. And what you can see here is that these regulatory T cells are mostly quite migratory, despite the fact that they see their cognate antigen, which in other contexts you would expect them to cause their migratory arrest. They form stable contacts with dendritic cells, for instance. But here they mostly migrate, and it is only in some areas, for instance, up here on the top left, or maybe here, where there's these areas where the cells need to stabilize the interactions. And by careful comparison of tumors which do express HA, these are these red lines here, or tumors that do not express HA, we actually see differences in the microbial group. And the parameter that we're using here is the so-called arrest coefficient. So that is a measure of the stabilization of cell contacts that T cells undergo in tissue. So a cell with an arrest coefficient of 1 is basically almost virtually immotile, versus a cell with an arrest coefficient of 0 migrates actively continuously without interruption. So you can see that most of the cells are somewhere in between here, and in the presence of antigen there's an increase in this arrest that the cells undergo. So we can actually see that this is an antigen-dependent arrest of the cells. So now what are the cells that present the antigen? For this we actually only had to wait a little bit longer, because the tritidextran has the feature that it leaks out of blood vessels into the tumor tissue, and then gets taken up by phagocytic cells in the tumor microenvironment. So these are macrophages, dendritic cells, and other cells. And what we find is whenever these regulatory T cells stabilize their immune behavior, it is typically here in the direct vicinity or in contact with one of those phagocytes that have taken up the tritidextran that has leaked out of the blood vessel. We know that it's a phagocytic cell, and we can define the locations in which this happens most frequently. In order to even more specifically identify the cells that present the antigen, we used a genetic reporter mouse. So this is a C11C M-cherry transgenic mouse. These mice express the red fluorescent protein M-cherry under the C11C promoter, which is a gene that is expressed by dendritic cells typically. And we can identify the types of cells that express M-cherry. So these are flow cytometry dot plots, and we know that the cells have certain characteristics that we can define. And now by imaging, we can also see that these interactions that Treg undergo in the tumor tissue environment is specifically with those tumor-infiltrating CD11C positive cells, which actually we probably wouldn't call dendritic cells. People now tend to call them tumor-associated macrophages, rather CD11C positive tumor-associated macrophages. So we have basically identified a specific cell population that presents antigen to these tumor-infiltrating regulatory T cells. And the only way that we could have achieved this, obviously, is by doing these dynamic imaging studies in the tumor. So beyond just looking at the behavior of cells and making inferences of what kind of signals are exchanged between these cells based on the degree of contact stabilization, we also wanted to visualize the signal transduction in these T cells directly in vivo on the single cell level by multiphoton microscopy. And this is just a very reductionist scheme of information flow during T cell antigen presenting cell interactions, where antigen in the context of MHC molecules binding to TCR, the T cell receptor, triggers a variety of signaling cascades which then converge on the activation of several families of canonical transcription factors. So the NFAT proteins, NF-kappa B proteins, and the AT1 heterodimeric protein families. And one of these, at least NFAT, is very, very characterized in that it resides in the cytoplasm of the resting cell. But when the TCR is triggered and it produces a calcium signal in the cells, NFAT becomes dephosphorylated and translocates into the nucleus, where it then binds to different regulatory regions and promoters of genes that are under T cell receptor control, and then drives different differentiation phase of T cells. So we thought that we could visualize maybe the translocation of NFAT from the nucleus, from the cytoplasm to the nucleus, and use this as a surrogate indicator of gene activation by NFAT in these cells, in response to the cells seeing antigen. And Anjana Rao, a collaborator of ours a couple of years ago, has made a genetic reporter construct, which is a fusion of GFP and the regulatory domain of NFAT. So that is the site where all the serines reside, which are dephosphorylated when NFAT is activated, and then exposes a nuclear localization sequence that drives NFAT in the nucleus. So our prediction was that when a cell would engage in a resting state and now engage with an antigen-presenting cell of any nature, a tumor cell, dendritic cell, whatever, that NFAT would then move into the nucleus, and we would be able to determine that this cell receives a productive T cell receptor signal. And in order to quantitative or semi-quantitatively actually monitor this, we could express a second nuclear counterstain. This is now H2B, the histone protein, but fused to MRFP. So here in the absence of antigen, we see the red nucleus, and we see the cytoplasmic NFAT-GFP report. And if we expose the cells to antigen, NFAT moves in the nucleus, and we have this overlap which we can quantify. This is NFAT signaling index. So we first validated this report in an imaging model that we have a lot of experience with. This is the proprietary lymph node. So here in anesthetized mice, we make a small skin incision behind the knee of the mice to expose the proprietary lymph node. And these anesthetized mice, as I said, and then we have visual access to our multiphoton microscope to this lymph node here with intact blood perfusion, innervation, and also efferent and different lymph flow of these lymph nodes intact. And we can again collect movies from these lymph nodes. And this is just an overview basically of such a lymph node here. The lymph node is maybe altogether this big. So this is just one section of the lymph node. And at this resolution, you will not be able to discern much detail, but this is just to show that these T cells here, those yellow objects here, because they co-express GFP and RFP, form stable conjugates with these blue objects, which are B cells, pulsed with the hemagglutinin antigen. So these cells here, these are basically antigen-driven stable interactions between B cells and T cells, whereas control B cells here, in white, don't form these types of interactions. So at higher resolution here, this is now a T cell where all the NFAT is in the cytoplasm as predicted. The nucleus is red, and this cell is going to engage with this antigen-presenting B cell in about a few seconds. So when I play the movie, you'll see that as soon as the cells make contact with the antigen-presenting B cell, NFAT GFP starts to move into the nucleus and then stays there for the duration of the movie. And we quantified this for many events, and typically it does not take longer than a minute and a half to 2 minutes for all detectable NFAT to the nucleus. So it's like a genetic on switch that is triggered and fully flipped as soon as a T cell engages with an antigen-presenting cell. We also looked at what happens during upon disengagement of the T cells from these antigen-presenting T cells, and that's shown here. So here you have two T cells which are about to disengage from these antigen-presenting B cells. And the interesting thing is here that even upon disengagement, NFAT remains in the nucleus for some time, for much longer than it takes the T cells to move all of the NFAT into the nucleus. So there's this asymmetry in the export. The microphone I can't turn on.
Well, just turn the speaker down.
Okay. So we can basically also quantify the export kinetics of NFAT from the nucleus, and what we've done now is to do studies how this influences gene expression of NFAT-dependent genes, these different import and export kinetics in the context of different types of cellular interactions. And obviously, we are interested in what the significance of this is for our tumor infiltrating T cells. So we again did studies here in dorsal skin fold chambers. In this case, we did not visualize the regulatory T cells, but we stained the T cells with the two genetic markers, the NFAT and the nuclear marker. And this is just an example of how this now looks. So here you have a tumor in the dorsal skin fold chamber. Here you see that the tumor cell nuclei. You can actually see some signs of tumor cell destruction here at the perimeter. So these are tumor cells that are being lysed by a mechanism that we don't fully understand yet. And you see T cells in different states of activation with regard to NFAT. So many cells are not activated, especially here in the tumor stroma, although you actually find some evidence of activation there too. And here in the tumor parenchyma, you find all kinds of states. You find stable arrests, but you also see T cells which continue to migrate while NFAT is in the nucleus. And we think that this prolonged export of NFAT is what is causing this type of event. But I don't want to go into this too much, just maybe to show you how we quantify this. So here we are really over time plotting the instantaneous velocity of these cells. So here these peaks here, these are peaks of migratory activity. This is a cell that is pretty well arrested and has a continuous NFAT activation state. So red color here indicates nuclear localization of NFAT with a high NFAT signaling index. Whereas these cells you have low NFP indexes, so those have primarily cytoplasmic NFP. And what's interesting here is that, again, what I described, there's these states of motility while NFP is active. And this is just a control tumor, so here we don't have expression of hemagglutinin in the tumors, and obviously as a result we don't have NFP activation, and the cells tend to migrate even more freely than with the present approach. So that's what I wanted to talk about, to say about signal transduction in T cells. And now for the final part of my presentation, I will talk about HIV infection and how T cells behave in these HIV-infected organisms. So one major question in the HIV field is how HIV actually spreads between the cells that it can infect, both between T cells, but also between some dendritic cell subsets and also macrophages which can be infected in humans, as you know from various studies. And there's obviously the idea that free virus gets released from cells and then binds to target cells. There's also the idea that free virus is basically taken up by dendritic cells and then presented in unprocessed fashion in infectious form to T cells. Macrophages can produce virus that then gets presented to T cells. But also the idea that T cells, infected T cells and uninfected T cells can directly interact, and that in the context of these so-called virological synapses that form at the interface, virus is transferred. So none of this has been actually explored in vivo. Obviously, humans are not amenable to experimental studies, and the different and monkey models that are susceptible to HIV infection are also difficult experimental models. So we thought we would start to look at these things in humanized mice and to address some very basic fundamental questions. So how does the migratory property of HIV-infected cells change compared to uninfected cells? And how are possibly the interactions with other immune cells changed when they're infected with HIV? And how is this occurring in vivo? So again, regular mice can't be infected with HIV, so we had to use very special mice. These are immunocompromised NOD mice with a SCID mutation and in some cases a gamma chain, cytokine gamma chain deficiency, which were irradiated and then transplanted with human fetal liver and human fetal thymus tissue and transfused with human hematopoietic stem cells. And over the course of the next few months, these mice reconstitute with a peripheral human immune system that is not fully functional, but that has many important functions of a human immune system recapitulated, and importantly, which can be infected with HIV. These mice here, after 3 to 4 months, we call them BLT, bone marrow, lymph nodes, thymus, humanized mice. In order to visualize the spread of infection, we used a special recombinant HIV strain of viruses, which generates a transcript. So these are the open reading frames of HIV. So here this is the open reading frame for NEF, where downstream of NEF an official iris GFP supporter is inserted. So whenever an infected cell starts to express NEF, it will also start to express GFP and become green fluorescent. So here again, so you have a virus that binds to a target cell, delivers its RNA genomes, which is reverse transcribed, integrated into DNA that integrates into the genome of the host cell. And once the host cell then starts to produce HIV proteins that lead to the production of new viruses, at the same time it will also start to produce GFP, rendering the cell green fluorescent, so we can visualize it. And the way we infect the mice is that we inject HIV GFP, this reporter virus, into the footbed because we want to visualize it in the draining propigeal lymph node, for which we have a model system established. And this just shows that we can also generate systemic infections with this virus. So here this is basically a non-fluorescent virus versus the fluorescent virus, and these are RNA copies in the blood of these mice or in the plasma of these mice. So within 1 to 2 weeks, we typically get robust plasma viremia indicating systemic infection of these animals. So here, this is flow cytometry, just showing that we see GFP positive events in the draining lymph node, whereas in remote lymph nodes we don't see this, so this infection stays local during the first week. And these infected cells are, as far as we can tell by flow cytometry, basically exclusively T cells, at least during the first few days. So these are CD3 positive human T cells, and you can see, for instance, that they downregulate CD4, which is a hallmark of HIV infection of T cells in humans as well. So again, I've already shown you this. Here we're looking at the complete lymph node, which is exposed by a small skin incision in an anesthetized animal. And then we can record over the course of several hours, up to maybe 10, 15 hours, if you want to have spend a long day on the microscope, our movies. So this is now an intravital micrograph from such a lymph node. Here you see the entire lymph node. This is a part of the cortex of the lymph node. Here's a piece of the so-called subcapsular sinus, which is where the lymph fluid that drains from tissues arrives. And that's probably where the virus also initially enters. You can see that these green fluorescent objects here, which are all T cells, have spread pretty homogeneously within the lymph node cortex, indicating that they have some means of getting around to distributing the lymph node. And when I play the movie, you can see that this is because these cells actually retain their motility. They migrate a little bit more slowly than uninfected T cells, which is something you would notice if you have seen a lot of these types of movies. So they migrate about half as fast, and you also probably notice that there are some of these cells have these very peculiar elongations here. Right? About a fraction of 10 to 20% of the cells that show this behavior. The others don't show it, but only migrate more slowly. And here again, we quantify this using, first of all, our arrest coefficient. So here these are basically cells which show increased degree of arrest compared to uninfected T cells. And we also see these elongated cells, which we quantify through the mean cell skeletal length. So the average mean skeletal length of these cells. So each dot here represents a cell that has been tracked over maybe 10, 20 minutes or so. So we were wondering what causes these elongations, and so we took out different viral genes, and one gene that we took out are the envelope genes. So envelope genes are, as the name suggests, sitting at the envelope of the cytoplasmic membrane envelope of the virus that it takes with it from the host cell where it is produced, and these envelope proteins are inserted. And these envelope proteins actually serve like adhesion molecules. They also have fusion molecules. They cause the virus to fuse with the target cell, but they also form adhesive interactions. And interestingly, if you take envelope genes out, these elongations completely disappear. But what does not disappear, and which is evident here, is the cellular arrest, the increased cellular arrest of these infected cell populations. So other proteins, apart from envelope proteins, are responsible for this deceleration. And right now we are trying to figure out exactly which proteins these are and for what purpose also. So another question, obviously, is so we know that envelope genes are responsible, but nevertheless, what causes these elongations? One idea is that via envelope, HIV infected cells can bind to CD4 positive cells. CD4 is the protein to which envelope proteins bind. And this could be a tethering, because the cells still continue to migrate, and could be, then lead to this elongation of the urothel. What is an alternative possibility is that cells fuse as a result of expressing N with unaffected target cell, producing multinucleated syncytia, which are then remain connected via these membrane bridges. And to discern between these two possibilities, we've generated another reporter virus, which is the same one as I've shown you before, but now here there's a nuclear localization sequence inserted just upstream of the GFP gene. So this causes the GFP, as soon as it is expressed, to localize into the nucleus. And the idea is that this would allow us to actually visualize the nuclei, and if we have multiple nuclei in these structures, we know that they're syncytia. If we only see one nucleus, we would know that this is more likely a tethering nucleus. And the result was quite clear-cut. So here, if you just look in the top left, that's the original recording of one of these movies. You see that there's this cluster which comes in onto the top right, and then all of a sudden disperses. And you can actually see these bright zones here, which are the actual nuclei with the highest expression of GFP, and there's 4, or actually I think 5 in this case, discernible individual nuclei that become visible as soon as this cluster here disperses. So here, basically, once the nuclear cluster, it looks like one big cell blob, and then it disperses. And here we're just rendering the brightest compartments of these cells in red, so we can actually see the individual nuclei better. And this is just another example of this. But this is not the only explanation. So here, these are basically structures with more than 2 or more nuclei. These make up most of the extremely elongated cells. This is against instantaneous cell skeletal length. But there's also some events where cells elongate, although they only have one identifiable nucleus. And this is here an example of such an event. So here you have this extremely long cell tether. Maybe it's better to see it here. But at the end of the cell tether, which is highlighted here by the circle, there's not another nucleus. So we suspect at this point that this could be one of those tethering events. And obviously, one question is, are these tethering events maybe the in vivo equivalent of these virological synapses by which T cells can transfer virus between each other? All right, so this is actually everything I wanted to say, and I hope everything came across clearly. Please let me know if you have any questions.
Yeah, so one question I had for you, Torsten, was in your experience, because you've done a lot of different kinds of imaging studies, not only in vivo studies, but the in vitro studies, within the context of immunological studies, what's been your impression of how in vitro findings compare to what you're seeing with in vivo work?
Yeah, so it's so many of the concepts that have been predicted based on in vitro studies of the immune system, especially in vitro imaging studies, have been in principle confirmed by in vivo imaging studies. So I would not say that in vitro studies are generally tend to be misleading or so, but the way processes play out in vivo tends to be different. Simply because of the much more complex tissue architecture compared to in vitro culture systems where you only have 1, 2, or maybe 3 cellular players united. So you basically see quantitative relationships. You see things play out in different ways in vivo, and often you see behaviors that actually were not predicted from in vitro studies. So you see complexities that reveal characteristics of the immune system and important regulatory processes that cannot be discovered and cannot be replicated in vitro. So I think this is where much of the value comes in. Also, if you do in vitro studies, you always have to make assumptions on how to best replicate the in vivo situation, because that's what you ultimately want to model. And you never know for sure whether these assumptions are correct in terms of, for instance, the stoichiometry of T cells and dendritic cells, or the specific type of T cell or its activation state that you generate when you culture these cells. To what extent these assumptions that you replicate the in vivo situation is always unclear. So there's always residual uncertainty as to whether what you find is real or not. And I think that's an important point to consider when evaluating the value of these intravital imaging studies, that it sort of presents an ultimate reality check to the extent that you can generate a model system which really behaves, replicates physiology.
Okay. For people that may not have had an opportunity yet to use multiphoton intravital microscopy to study immunological problems, what would be your sort of advice to somebody that thinks that they have some interesting questions they might want to start using this technique to get engaged in their research?
Yeah, so I think in order to successfully use this technology, the best way is to start a collaboration with somebody who has a lot of experience with this technology, and that basically professionally uses this type of approach. And try to engage oneself as much as possible with the actual conduction of these studies in order to learn of the ins and outs. And if that, in the context of that collaboration, one finds out that the approach really helps you to advance your own science, at this point you can make the decision whether it is worth it to actually invest in getting the system up and running for yourself. Because from the time point that you make the decision until you get your first movies and your first useful actual data out of it, there's quite a bit of a learning period. So in my experience, it takes probably a year to a year and a half from the time point that you actually start to do these studies yourself until you really can produce reproducible data of interest for yourself. So it's an investment not only in terms of purchasing a microscope, but also investing into the infrastructure, the logistics that are involved with doing these kinds of studies in terms of mouse models and so forth. So that determines whether you would want to make this decision or not.
Great. Okay. So are there any other questions from the audience at this point? Okay, well, if there's no other questions, we'd like to thank you for your attendance today, and I'd like to remind you that you'll get an email after the webinar is done that will provide you with a link that you can review the webinar again. If you have any further questions at a later date, please feel free to email the questions, and we can review them later and provide you with answers. Thanks again very much for your attendance today.